Hello, everyone. Thanks for coming to our news talk every Tuesday. So today we have three excellent speakers on the topic of human-induced pluripotent stem cells. I am the moderators. I'm Zheng Ma. I'm an associate professor from biomedical engineering in Syracuse University. And so today's overall topic is the human iPSC technology shaping future biomedicine. We have three excellent speakers. The first one is Zheng, who is an assistant professor in Syracuse University of Biomedical Engineering. Next speaker, Xiaoping Bao, is an assistant professor from Purdue University, Chemical Engineering. And our last speaker, Huaxiao, he also assistant professor of biomedical engineering from University of North Texas. I'm going to give a more detailed introduction of each speakers right before their talks. And we also have a panelist, Jing Guo. She have her PhD degree in Washington University at St. Louis, and now she's a postdoc fellow in the University of California, Berkeley. So today's presentation will be in the order, start with Dr. Zheng, and then Dr. Bao, and then Dr. Yang. And after that, we will have a 30 minutes discussion. Now, let's welcome Dr. Zheng. He is a now assistant professor in Department of Biomedical and Chemical Engineering at Syracuse University. He received his PhD in mechanical engineering from University of Toronto back in 2014, and he has his bachelor degree in Zhejiang University. Before he joined Syracuse University, he was a postdoc fellow and a research scientist in University of Michigan Ann Arbor. In Jianping Fu's lab. He has published over 30 papers in high-impact journals, including Nature and Cell Stem Cells. Today, his talk will focus on embryonic sac embryos using human pluripotent stem cells. Now I'm going to stop sharing. Dr. Zheng, you can share your slides. Yeah, please.
Okay, thank you very much, first of all, for the introduction. It is my great pleasure to be here today and to share some of my research. The topic of my presentation is a microfluidic embryonic sac embryoid using human pluripotent stem cells. The embryonic development is undoubtedly one of the most amazing events in the biological world. Given the complexity of the organisms, it all starts from a fertilized egg or embryo. So this video is showing how a frog embryo, through embryonic development, turn into a living tadpole and then a frog. So every time when I watch a video like this, I may take a moment to enjoy its beauty. And at the same time, I can't resist wondering whether there is any hope for us to understand the secrets behind this process. So here I quote a famous saying from one of the earliest American developmental biologists. The greatest of all wonders of the material universe, the existence in the simple, unorganized egg of a power to produce a definite adult animal. So developmental biology is actually a very old topic. For more than a century, a lot of developmental biologists have been using different animal models to study embryo development. So most of our knowledge about mammalian embryonic development is from studying mouse. Why we study mouse? Because they are easy to access. But the morphological and the genetic differences between these animal models and humans make cross-species comparison very problematic. So this is an overview of mouse and human development. Now let's zoom into the stage around implantation. Now, as you can see, before implantation happens, human and mouse embryos, they appear structurally similar and contain the same cell types. But shortly after implantation, the two embryos, human and mouse, they become very different. So they also contain different types of cells. And based on the drastic difference showing here, we know that the mechanisms revealed by studying mouse embryos may not completely apply to human systems. But the problem is due to the ethical issues and a lot of practical limitations. Doing experiments using human embryos, even with other primate, for example, monkey embryos, are very challenging. So here we use human pluripotent stem cells to build human embryo-like structures, which can offer a tractable platform so we can study this black box of human development. Now let's take a closer look at the human embryo right after implantation. So let's focus on the inner cell mass and the derivatives. So all the extra embryonic tissues are shaded in this cartoon. So the first, so we start from a clump of pluripotent stem cells. And the first major morphological milestone of an implanted human embryo is the apical basal polarization of the epiblast and the formation of a pro-amniotic cavity. So this process resolves the epiblast into an asymmetrically patterned embryonic sac with squamous amnion tissue at the dorsal pole and the columnar epiblast at the ventral pole. And the formation of this amniotic sac heralds the onset of gastrulation. And the human human primordial germ cells are also specified in this process. So the pluripotent stem cells we're using, if we want to define their developmental stage, they roughly equal to the cells in this between this stage and this stage. So if we look at the morphology, so in order to form this asymmetrically patterned amniotic sac, so one side of the epiblast differentiates into amnion tissue, but the other side remain pluripotent. So we figured if we want to model this process, we can have a cluster of pluripotent stem cells and apply the amnion inductive signal to only one side of the cluster. So that's the main idea of the microfluidic device design. So here is how we did it. It's a very simple microfluidic device. So it had 3 parallel channels, and they are separated by evenly spaced supporting posts. The gel, in this case, we use gel tracks, were injected into the central channel, and the gel tracks will contract during gelation to form these biconcave shape gel pockets. And then suspension of pluripotent stem cells will be injected into the cell loading channel and allow to settle into the gel pockets and to form cluster. If we zoom into the gel pockets, right after the cell seeding, they just randomly random cells in the pockets, and then they will form a cluster, and they will go through luminogenesis. If we don't do anything, we do not apply any inductive signal to the device. So here is the control condition, where we just let the cluster of the cells to grow. So they form a central lumen enclosed by pluripotent stem cells. they are positive for all the pluripotent markers. Well, these bright field images are showing the initial cluster and the lumen after 36 hours. So this setup allows us to stimulate only a part of the cluster by applying the morphogen or inductive signal to only one side of the device. So this video is showing how the pluripotent columnar cyst is forming in the micro device. So these clusters are from the same microfluidic device. Now it's time to add an inductive signal into the system. Of course, we tried several growth factors, which are very well known, are involved in human development. In this case, we apply BMP4 because it generates fantastic results. This is a time-lapse video showing the progressive development of the embryonic-like sac in the micro device. We can see the formation of the central lumen, and the cells exposed to BMP directly will differentiate into squamous amnion cells, while the other side eventually will migrate out from the structure, causing the collapse of the embryonic sac. And before the structures collapse, we stain some known markers. So TFAP2A is the amnion marker, and the T is a primitive streak marker. We see the symmetry breaking of the structure is very obvious in this case. And the CDX2 is a marker for both amnion and the primitive streak. So a good thing about this device, it is very reproducible. So now what we are seeing is an array of the cluster. They're showing synchronized development. We see the morphology, the timing, they are almost the same. And not only the morphology is the same, but also the markers. Here are the same devices, same clusters showing in the previous video. Again, TFAP2A is the amnion marker, and T is the primitive streak marker. So everything looks very nice. And because of this reproducibility and controllability, so some public media call this system mass produce or mass manufacturing of human embryos. I personally am not a big fan of this term because it's implying that we are making like humans or mass manufacturing humans. But indeed, if we want to make a lot of them, we can easily make a lot of them. As I mentioned, there's a very important type of cells which are specified during this process, which is the primordial germ cells. Primordial germ cells are the embryonic precursor of sperm or eggs. They transmit genetic and epigenetic information across generations. And more importantly, the understanding of the development of primordial germ cells, or PGCs, is very critical for reproductive medicine. Some very new study showed that very different from other animals, human or primate PGCs are specified in human amnion. So then the question is, if we want to model this amniotic sac development, whether we have these germ cells or where are the germ cells emerge. So these are two immunostaining images. So the top is from the monkey embryo. As I keep saying that we don't have access of implanting human embryos, so we study monkey embryos. And the bottom is from our amniotic sac model. TFAP2C and SOC17, they are a germ cell or PGC markers. So based on the two sets of immunostaining images, we see not only the morphology, the shape, they look almost the same, but also the specification of germ cells. They are consistent with our observation from monkey embryos. Again, everything is very reproducible and controllable. So if we stain this array of embryonic sac, so all the embryonic sac contain the germ cells, and we see germ cells in the amnia. So everything is consistent with monkey embryo observation. So Wnt signaling is known to be important in germ cell development if we apply a Wnt inhibition into the system and all the germ cells is gone. So this is superior controllability is very good for further mechanistic studies. But which makes our presentation of the data a bit difficult sometimes, because the reviewers or readers want to see. Usually, we have the average and a standard deviation or standard error of mean, so you can calculate the p-value. But in our case, if we apply Wnt inhibition, it's zero. So none of them has PGCs. So we don't have the bar plots for this kind of study. And of course, now I just have shown you the staining of some known markers. But at this very early stage human embryo development, a lot of markers, they overlap. A single marker or just a combination of 2 or 3 markers sometimes is still not efficient to convince people that we get the right cells. So here we validate our model using single-cell RNA sequencing. So basically, we dissociate the cluster of cells into single cells and sequence individual cells. So in the end, we can obtain a matrix of a gene expression of individual cells. This is a high-dimensional database, which can be visualized using dimensional reduction and unsupervised clustering. So in these plots, simply speaking, so we use published human embryo data to compare with our data from the amniotic sac model. In this plot on the left, so all the color-coded cells are from humans. The cell lineage or cell types are labeled here. So in the plot on the right, so all the color-coded cells are from our device. The understanding of these kind of plots is if the cells cluster together or they show together in this plot, and that means they have a similar transcriptonic profile, and we can claim they are the same type of cells. And as shown here, so our amnion-like cells cluster with human amnion, our PGC-like cells cluster with human PGC, our mesoderm-like cells cluster with mesoderm, and the epiblast cells cluster with human epiblast. So everything is a match. So now we can confidently claim that we actually have a very realistic model that can help understand this very early stage of embryo development. So now we have a good model, then what we can do about it? So here, there are many things, but here I will give you just 2 or 3 very simple examples. First, we can do live cell imaging, which you cannot do it in the real embryo, especially during the replantation. So the plots on the left is the staining of the primitive streak marker, T. Very interestingly, we observe that T always emerges at the junction of amnion and epiblast, and then propagates to the rest of the cyst. So these are the snapshots because we're doing immunostaining. But with this model, we can generate a T reporter stem cell line and to visualize this dynamic process. This plot is showing the reporter signal, the co-staining of reporter signal and the T antibody, just to prove our reporter is actually working. And in this video, we can clearly see the emergence of E positive cells is from the junction of amnion and epiblast. So this is a very interesting observation. And then you may ask, why the primitive streak have to initiate at the junction of amnion? If you think about the real human development, it's actually a very interesting interlock mechanism, which can give you or guarantee the correct developmental sequence. Right? In the real human embryo development, you always have amnion first, and then primitive streak. It's never primitive streak comes first. That's why we see this a very interesting observation in our model. So based on these observations, we came up a hypothesis that the amnion must be signaling the epiblast to differentiate into primitive streak. Otherwise, how they control this sequence? So in order to validate this hypothesis, we design a very simple experiments, which we use transwell. The reason of using transwell is just we want to make sure all the cells have enough access to BMP so they can differentiate into amnion. So in this case, we differentiate all the pluripotent stem cells into amnion first. We confirmed by the marker. And after we have amnion cells, we're just going to seed undifferentiated pluripotent cells into the same well. And in this culture, we do not add anything. We don't add BMP, Wnt, or anything. But after 1 or 2 days of co-culture, all the pluripotent stem cells, they turn into a primitive streak phenotype, as indicated by the T staining. So this simple experiments prove that amnion is actually functioning as a signaling center to control the emergence of a primitive streak. So single-cell RNA sequencing is a very powerful tool which gives us the transcriptonic profile of individual cells. That is a lot of information that we can leverage for more discoveries. So in this case, we conduct the single-cell RNA sequencing for the model on 24, 36, and 48 hours. You can see their morphology. And we get the data from each time point, and we can integrate them here. So in order to see how the lineage separates from each other. So now here we see a developmental path from epiblast to primitive streak and the mesoderm to amnion, but we can't see a clear developmental trajectory for the germ cells. So we figured that, so now in here, we are visualizing the data, the high-dimensional data in 2D UMAP. Maybe some information is missing. The algorithm doesn't think this information is important, but in reality, from developmental biology perspective, it is actually very important. So we use another way to analyze the data, which is not a very standard way for analyzing single-cell RNA sequencing, which is called the diffusion map. So in this plot, we see that all the lineages display distinct and well-separated trajectories. So the yellow color is the pluripotent stem cells, which is a starting point, which the cells go to amnion, go to primitive streak, and then they show sort of like a vertical geometry in here, and it goes to PGC cells. No wonder if you project into them into 2D, this information will be lost. So this is like a rotating of the 3D plot, just we have a better visualization. With this plot, there are many things we can do. For instance, if we are interested in how amnion is developed, we just take out all the amnion-related lineages and analyze them again. These are pseudo-time analysis. We see how they change. And since there are two different mesoderm lineages, so we can see a branch of a primitive streak into mesoderm cell 1 and the mesoderm cell 2. So if we just take out the PGC, the germ cell related lineages, we see a very clear branching between amnion and the PGC. These are the RNA velocity, and we can identify a branching point. Like all these cells at this point is actually a common progenitor of human amnion cells and the PGC cells. So this is very important for human embryology or primate embryology, because I don't think if you remember, but the mouse embryo or even pig or rabbit, they don't have amnion at this early stage. And their amnion all appear after the germ cell specification. But in human or primate, it's actually coupled with amnion development. Since it's an in vitro system, our lab has planned to further probe this amnion and PGC bifurcation. It is really convenient because we can generate a polar line, we can do knockout, we do IDE inhibitor, so everything can be conveniently done. So another thing we can do with these models is we can analyze their cell-cell crosstalk, like who's sending out what and who's taking what kind of growth factors. In here, we grab some commonly known important pathways. For instance, BMP, Wnt, or nodal. So for BMP and the Wnt signaling, we see it's very, very complicated crosstalk. It's like everybody is sending out the BMP and everybody is taking BMP. But interestingly, for nodal signaling, only the primitive streak cells are taking nodal, not other cells. Well, inevitably, you will think like a nodal will be really important for primitive streak. In this case, if we want to validate this, we just generate a nodal knockout. We repeat the experiments. Of course, we observe very obvious phenotype. For instance, in the wild type, the primitive streak will start forming and start to become migratory. But in the nodal knockout model, so most of the cells become squamous amnion cells. We don't see cells migrating out. And the cell fate was confirmed by the staining. We see the amnion marker, SL1, GAT3, and the primitive streak marker. And the PGC's number is also affected in this case. Just a quick summary in the end of my talk. So we are really excited about this emerging field in the cross-section of stem cell and the human developmental biology. So right now, the name we call it called synthetic human embryology. There are really a lot of potentials on this. But there are also some limitations because in our model, as I showed in all the examples, we can do a lot of things, right? Real cell imaging, genetic manipulation, drug perturbation, whatever you want to do. But in order to validate these findings, it's kind of a challenge because, of course, doing experiments on human embryos, especially post-implantation, is illegal. Doing these experiments using monkey embryos also has a lot of challenges in terms of technology, and they have their ethical restrictions too. And also, if we have this, after we have this platform, we want to disseminate the technology to developmental biology groups and the stem cell research groups. But it involves some microfabrication and operation of the microfluidic systems. From my perspective, I was trained in mechanical engineering. It's not an issue, but for some biology lab, that may be an issue. So in the end, I want to thank, of course, most of the work was done in University of Michigan. Especially, I want to thank Dr. Jianping Fu, my advisor, and also the funding agencies. Thank you.
Thank you, Dr. Zheng. So I would like to give my special thanks for Dr. Zheng's very nice talk on early human embryo development. Now we can move to our next speakers, Dr. Bao. Dr. Bao, do you want to share your slides? Xiaoping Bao is currently the William Luco Assistant Professor in Davidson School of Chemical Engineering at Purdue University. He received his PhD degree in chemical engineering from University of Wisconsin-Madison in 2016, and before that he received his bachelor degree from Tsinghua University. After his PhD degree, he was a postdoc fellow in chemical engineering at the University of California, Berkeley. He published over 50 papers in high-impact journals, including Nature Medicine and Nature Biomedical Engineering. And recently he was awarded of Young Investigator Award of Cell Tissue Organ Journal, and also 2023 BMCE CNBE Rising Star Junior Faculty Award, NSF Career Award, and also NIH CATS Early Stage Investigator Award. So now let's looking into Dr. Bao's today's presentation, which is engineer human pluripotent stem cells for targeted cancer immunotherapy.
Thanks, Zheng, for the nice introduction, and also thanks, Dr. Zheng, for starting with a really nice talk. And give a lot of background about human pluripotent stem cells. So, unlike Dr. Zheng's talk, modeling early development, my lab more interested in terminally differentiated cell type for targeted cell therapy. So, and a brief introduction about stem cells. What you can see here is a cartoon showing there are in general two kinds of human pluripotent stem cells. One is a human embryonic stem cell, which isolated from development embryo at the early stage of development. And the second one, or the major breakthrough in the field, is the induced pluripotent stem cell technology, which is like a 2012 Nobel Prize in Medicine award, where a scientist called Shinya Yamanaka, who reprogrammed skin cells, or nowadays any somatic cells, back to the pluripotent stem cells, and these are called induced pluripotent stem cells. So whatever the cell you use in terms of HPLCs, they share two defined properties. One is the self-renewal, which in other words, they can produce unlimited cell source. And second, they have the ability to become every single cell in our body, including, and what is shown here, blood cells, cardiac cells, neuron, liver cells. So again, these are more differentiated, terminate differentiated cells for regenerative purpose. So these cells are like just like a human embryo model in vitro. These are human cells, which in general really tough to get from human patient samples. With these human cells in the dish, you can do lots of like application on these cells, including drug screening, disease modeling, cell differentiation, or cell therapy. So my talk today will focus on blood, or in general blood and immune cells, and also their application in cell differentiation and the cell-based therapies. So before that, I just want to give a brief introduction about the very popular nowadays in immunotherapy, the chemical antigen receptor, or short for CAR engineering. And you can see in this cartoon on the like my mouse arrow points, there's a CD19 positive tumor cells, which is larger, and underneath there's a T cells. And in the patient, most the patient immune system, including T cells, they cannot recognize the tumor cells as an antigen, so they kind of get fooled by the tumor cells. So in the past 20 decades, people in the immune engineering field have invented a technology called CAR engineering, where you can see they put a synthetic receptor, usually composed of extracellular binding domain using antibody-like structure, or SFE, and followed by some signal domains that are original in T cell signaling. So with these, they can specifically binding to the antigen on a tumor, So theoretically, any antigen can be bound by this CAR construct. An example shown here is the CD19 leukemia cells. So with this synthetic CAR engineering, the T cells can now recognize the tumor cells in the patient, and then they can activate the T cells by like a proliferation, release cytokine, or the general license the tumor cells. So these have been very effective in treating leukemia or many other blood disorders, blood cancers. And indeed, there are already 6 FDA-approved CAR T cell therapies as are listed in this table. And as I mentioned, the dominant approved cell therapy using CAR T cells are primarily on the leukemia or blood cancers. So the field is trying to move this area into more like a progressive solid tumor area. So in general, the standard pipeline to manufacturing CAR T cells, as I shown here, is the 6 steps. When there's a patient come to the hospital, the doctor or the clinical team, they will isolate the T cells from the patient blood. After that, they will isolate or purify T cells because the majority of the blood cells are like red cells or white blood cells instead of immune T cells. So after purification, they will then transduce the lentiviral construct, dominantly lentiviral or retrovirus. After that, they will expand the engineered CAR T cells. And they also need to isolate the final product with the successfully engineered CAR T cells. And after that, they will put it back to the patient. So these 6 steps, they have still the standard pipeline to manufacture CAR T cells in the immunotherapy field. However, there are currently many challenges associated with this pipeline. The number one is the cost is very high, or it's very very expensive process. And the cost can, on average, cost up to $500K per patient in the field. And as I briefly mentioned, the efficacy is extremely limited for solid tumors, including the very aggressive glioblastoma or short of GBM that I will talk later, where there's a blood-brain barrier. And there are a few other things, including primary T and other primary immune cells isolated from the patient or healthy donor. They are in general more resistant to genetic editing. And also, overall, this pipeline is very time-consuming, so it's not suitable for very acute conditions. So overall, there are many challenges associated with this promising technique. So as a stem cell bioengineer, we are trying to leverage the power of stem cell to address one or a few of these common challenges associated current manufacturing of CAR T cells. So what we propose here is kind of to generate more off-the-shelf CAR immune cells. This is not only including the T cells or CAR T cells. And what we did is listed here. We have the starter normal human iPSC cell line, either iPSCs from the patient or from a HLA matched donor. And then we will apply genome editing. A famous example, including here, is the CAR2 for Cas9 genome editing. After that, you will have a stable CAR HPLCs, as indicated by my mouse arrow here. So I put a Y struct here to indicate the successful integration of the CAR construct on the stem cell. So this is a stable cell line, just like a regular HPLC line or cancer cell line. They can expand on like limitedly, and they can become, more importantly, they can become every single like immune cells type in our blood. And by applying the direct differential protocol, you can convince these engineered CAR HPLCs to become CAR express HPLCs, hematopoietic stem and project cells, or short for HSPCs, and then become off-the-shelf terminally differential CAR T cells, CAR B cells, CAR natural killer cells, or my talk today, one of my interested cell population is the neutrophils. So with this pipeline, as I mentioned, you can make all kinds of CAR immune cells, and they can proliferate unlimitedly. And another advantage of this pipeline that I propose here is the CAR expression after genomic editing and a single cell clone isolation. You can have really uniform CAR expression of the stem cells, and also the immune cell product. Usually they are more homogeneous with the very like a same genetic background. And a second like advantage, which is really tough for like when people using primary cells, you can make a different combination of or cocktails immunotherapy with different CAR T, CAR natural killer, or other CAR immune cells that which again is very difficult to perform using primary cell source. So for the next like 15 minutes each, I will give you a few examples what have been done in my lab with this pipeline. So the first example, we want to engineer really off the shelf and also universal like CAR immunotherapy from human pluripotent stem cells. So what we use here is in collaboration with the Philip Lowe group at Purdue, where his lab invented anti-FITC CAR. So in general, FITC is a fluorescence like a dye that to label cells, but instead of direct target the tumor cells, we engineer the CAR T or here the CAR stem cells with anti-FITC SLV. So these CAR, they will only bind to the FITC, but not direct to the tumor antigen. So to bridge the gap between the tumor antigen and also FITC CAR, the Philip Lowe lab developed lots of bio-specific adapter. Show example here including FITC folate to target the folate receptor positive cancer cells, and DUPI, PSMA positive prostate cancer. So with this different like a small molecule, you can theoretically target all kinds of tumor cells with the anti-FITC CAR. So what we exactly did to stem cells, we directly knock this CAR construct that target the FITC into the AVS1 safe harbor. So this locus is very famous for transgene expression in stem cells. So after knocking, you will see by the flow plot with the dye staining, you can almost get 100% knocking of the FITC CAR after differentiation. And a brief like a license test here. We tested two different CAR. One is the breast cancer cell line MDA MB231, and the second one is the prostate cancer. So you will see the highest the highest bar here is always goes to the engineer with the FITC CAR, and also in the presence of the like a bio-specific adapter or small molecules. In other cases, they have only baseline killing of the natural killer cells. So same thing applied to a second tumor cells, prostate cancer, only in the presence of the right bispecific small molecule and the physical, you will see a boost of anti-tumor killing efficiency in the fetal lysis assay. So the other advantage of human stem cell engineering is you can kind of put all kinds of editing to the human stem cells. So build up on the physical, as shown on the right, on the fetal engineered stem cells. You can put additional anti-PD-L1, which is a very famous immune checkpoint therapy in the solid tumor field. But we engineer this into a CAR construct. But it's not only improve the tumor killing ability of the natural killer cells or immune cells. We also include mutate a STA T3 binding motif in the CD3 signal zeta signal domain. And the function of this YXQ motif is it can active more active the cytokine signaling in natural killer cells and promote natural killer cell proliferation and only when they bind to the tumor cells. So this will reduce the proliferation or overexpansion in the whole body of a patient, and they will only kind of proliferate when they reach or target the tumor cells. So we kind of call this as a dual CAR engineering in our stem cell engineering approach. And the first thing we want to validate the proliferation or the signal activation after engineering with this anti-PD-L1 CAR. And you can see when you put both CAR or the anti-PD-L1 CAR NK cells, you have more expression of phosphorylated ST85 or and phosphorylated ST83 signal domain, which will again the marker of cell proliferation and cytokine release signal in NK cells. And also we also co-culture cells with the tumor cells, and you will see in the presence of anti-PD-L1 CAR, you will have about 10-fold more of cell expansion as compared to single CAR, single physical or the wild type natural killer cells. So this data indicate the successful engineer of proliferated NK cells in the presence of tumor cell antigen. And we also perform flow cytometry analysis on the other two famous signal pathway, IFN gamma and CD107A. And you can see the dual CAR has the most active the natural killer cells when they meet the tumor cells. So the next thing we just move forward to put into a simple mouse model, where immune deficient mouse model will transplant with tumor cells in the kind of the stomach area, and then we apply our engineered NK cells by IV injection. So after certain days, we will collect the sample to for cytokine analysis and also for tumor killing ability analysis. And I'm not going to show all the results, but although here the measure the tumor size along with the treatment, and you can see what I point here is the individual tumor growth curve, and on the right is the kind of a quantification of the tumor growth with all kinds of different natural killer cells treatment. And as compared to the PBS or the negative control or any other single CAR, you will see the bottom line, the purple one, is the dual CAR engineered natural killer cells from human primary stem cells. And you can see they are the best one in slow down the tumor growth in the xenograft mouse model. So I think at least the first one example in our lab. And the second example, or more interested tumor type, is a glioblastoma, or short of GBM. This is really aggressive, like a tumor type in cancers, or in general, like brain cancer or all kinds of solid tumors. And you can see from the data, this GBM patient, there are not lots of patients who are suffering from gliomas, only like 70,000. As compared to many other cancers, this is a very relatively less population. However, it's a relatively aggressive, or one of the most aggressive one. Less than 10% of patient after diagnosed can live beyond 5 years, and most patient will die in about 20 months. So this is there are multiple reasons contribute to the aggressiveness of GBM. The first one, the standard of care, for example, surgery or chemotherapy, is in general very ineffective in the brain cancer, because the brain is a very fine structure, and any surgery will destroy the function or our health of the brain. And two, the blood-brain barrier, which is a kind of a natural barrier that exclude all the toxic from the bloodstream. They also preclude the transmission of emerging or very promising drugs or therapies, including the CAR T cells, which make the treatment of GBM really challenging. So, however, scientists or clinical doctors in the field, they are still trying to develop or invent new therapies to treat glioblastoma. And my lab is really, again, interested to using stem cell engineering to develop cell therapies to treat glioblastoma. And I'm not going to talk again the CAR T construct here, but I just want to highlight in terms of the CAR T therapy against the GBM, there are already over 18 clinical trials ongoing. And what I listed here is the a clinical trial number, if you are interested to check. And above that, highlighted in red, is the antigen that targeted by these clinical trials. And I will use two of the these a CAR construct or target antigen. One is the IL-13 a receptor alpha 2, and the other is the MMP2, where targeted by colon toxin. So this is a 36 amino acid peptide that can isolate from scorpion venom, and they can specifically target the antigen of MP2 on GBM cells. So similar to what we did to the universal physics CAR, we kind of designed 3 different constructs based on the IL-13 and also chlorotoxin, or short for CLTX. So based on the signal domain, we designed 2. One is a specific for T cells. We named the chlorotoxin T-CAR. The other is CAR natural killer cells, where you have specific transmembrane of NK cell signal domains, and also more NK favor or specific co-stimulatory domain. And for the IL-13 CAR, we only use the T-CAR for the construct. So this kind of the different name of this construct. And the same thing, we just apply CRISPR-Cas9 mediated genome editing to knock this construct into the AVS1 locus of stem cells. And after that, you will see even differentiate into update 21. You will see very homogeneous and stable expression of the CAR construct in all kinds of the CAR engineered stem cell line. So the first thing we again first apply these cells, CAR engineered stem cell, to make CAR natural killer cells, and you will see the chlorotoxin natural killer CAR performs the best in killing the tumor cells in the dish. And this improvement is only like applied to the glioma or brain tumor. Other tumor cells, they don't, they only have the baseline killing efficacy. And also, more importantly, they are killing very minimal, minimally against the original or healthy H9 cells. And we did a very kind of preliminary like in vivo study where we put natural killer cells engineered or with or without a CAR construct and control compared to the PBS control. And you will see while the CAR engineered NK cells did the best job in slow down the tumor growth in the tumor bearing mouse, but they still have some variability, or they are not eliminated the solid tumor cells, which again highlights the challenge or the barrier of blood-brain barrier in treating glioma. So we are trying to seek if there are any other cell types in the immune system that can be harnessed to target the glioma. So what first come into our mind is the neutrophil, because they are the most abundant white blood cells, and they have the ability to cross any barrier in our body. So we just reapply again the engineered cells into a different differentiation protocol to make a neutrophil. So we also did the kind of cytolysis assay in the dish, and surprisingly, as you can see in the light, green line called toxin T CAR neutrophil actually behaves best against the other cell, CAR neutrophil differentiate from different CAR stem cells, and they even did a greater job than CAR natural killer cells. As you can see from what we mix different ratios from 100% neutrophil to 100% natural killer cells, the CAR neutrophil did a much greater job than CAR NK cells. And unfortunately, we didn't see any combinatory effects when we apply both neutrophil or NK cells. So we then next motivated by promising in vitro data, trying to test the function of CAR neutrophil in mouse model. And the first thing we're trying to do is we want to see if the neutrophil from human stem cell they are truly moldable, and they can penetrate any tumor or any site in the mouse. So we labeled the neutrophil or CAR neutrophil with a CY5 dye, and after that we will image 1 hour, 5 hours, and 24 hours after injection. And you can see in 1 or 5 hours, the cells they can migrate across the whole mouse body, and there's no huge difference in different neutrophil. However, after 24 hours, you can see more cells or neutrophil were enriched in the brain compared to most majority of unengineered neutrophil was stuck in the stomach area. And this quantification, you can clearly see more cells after CAR engineering. Neutrophil will migrate to the brain. That's kind of motivate us to test the anti-tumor killing effects of the CAR engineered neutrophil. So the same model, but we apply the different engineered CAR neutrophil, CAR NK cells. And I'm not going to explain more here, but just want to highlight the red one where you have the CAR engineered neutrophil. They did the best job in slow down the tumor growth, although you can see there was still some residual tumor cells in the mouse, but they actually did the best job in slow down the tumor compared to the CAR NK cells. And there are some interesting observation where you can see unengineered neutrophils or primary neutrophil or stem cell derived cells. They promote the tumor migration outside the brain. So this is a very interesting phenotype, and demonstrate the plasticity of the neutrophil. So what we quantify here is the tumor growth curve, and you can see CAR engineered cells was the best in slow down the tumor growth. They're also the best one in extended life shelf of tumor-bearing mouse. And one kind of more fundamental study is actually CAR engineer actually sustain the anti-tumor N1 phenotype in neutrophil. As you can see, unengineered neutrophil, they promote the tumor migration into other organs. However, the CAR engineer one, most of the cells will stay in the brain. And we also did a very brief RT-PCR to show CAR engineered cells actually express more of the N1 phenotype genes. Due to the a limited time, I'm just skip these. We are working trying to using the single cell RNA-seq to elucidate the underlying molecular mechanism. And I will just quick skip here that in combination therapy is also possible, where we loaded a nanoparticle chemotherapy drugs into the CAR neutrophil to make a combination of immunotherapy and chemotherapy. And at least kind of the successful loading of the drugs into neutrophil, and then nothing changed in migration or in the behavior. But after loading the drug, you will see much better anti-tumor efficacy. And we did the same in vivo mouse study. And I just want to highlight three. Whenever you have a combination, you will significantly improve the lifespan of tumor-bearing mouse as compared to the single like a CAR neutrophil or single drug treatment. And I just cover is using mouse model, and we are actually moving to using more like a big or large or more like a real patient, the pet dog patient. And we tested the toxin CAR neutrophil human one against the canine glioma cells, and they indeed still present some tumor killing ability. And we already performed safety in the healthy dogs, and they are very healthy. That we just got the data, and I'm not going to show here, but just want to highlight. Even you inject multiple times of a human CAR neutrophil into dogs, there's a really very healthy in general. So just conclude my talk today. I hope I convince you by this talk that we can engineer off-the-shelf CAR NK, not a T cells yet here, for target immunotherapy. And surprisingly, CAR neutrophil present a really superior anti-tumor efficacy. And also you can combine all kinds of therapy with the neutrophils. And also, we are trying to move to animal clinical trials into dog models. And the safety study is already done, and we kind of now recruiting the a pet dog patient for animal clinical trials. And with that, I'd like to thank her, my talent postdoc, Dr. Chang, who did most of the work for my presentation talk today, and also many collaborations across Purdue or other university, and also funding source from NIH and NIST. With that, I will be happy to take any question after the third talk. Yeah, Zheng, you are muted.
Thanks. Thanks. I was just saying, thanks for Dr. Bao for such a nice presentation on iPSC-based cancer immunotherapies. And now we already move to our third presentation from Dr. Yang. Dr. Yang, maybe you can start to share your slides. So Dr. Huaxiao Yang is an assistant professor at University of North Texas in Biomedical Engineering Department. He graduated with bachelor degree in chemical engineering from Huaxiao University of Science and Technology, and with a minor in biology science from Wuhan University. He got his a master's degree in micromolecular science from Fudan University, and then PhD degree in bioengineering from Clemson University. And then he worked as a postdoc fellow in Joe's lab at the Cardiovascular Institute, Stanford University School of Medicine. So he published over 40 peer-reviewed papers, including high-impact journal of Nature Cardiovascular Research, Circulations, and PNS. Dr. Yang was a American Heart Association postdoc fellow, and also a UNT Washington D.C. fellow, and a mentee of AHA BCVS mentorship program. Now let's looking forward to his talk on modeling cardiovascular development and disease with human pluripotent stem cells in 2D and 3D models.
All right, good afternoon, or good evening, good morning, everyone. So thanks so much for the invitation, and thanks, Dr. Mar, the introduction. So it's pretty honored to be here and also present. And also, we are talking a lot about pluripotent stem cells. So also, we are doing the same thing, but also more towards into the cardiovascular developments and also disease in 2D and 3D models. So first, I want to introduce my group. We kind of pretty diverse group from 5 different directions to study the human cardiovascular development disease. We're using human-derived organoids and also cell stretching, mechanic biology, machine learning, organoids on a chip, and also hydrogel biomaterial studies to achieve the goal. So overall, we would like to study the multi-scale cardiovascular tissue engineering from nano, micro, single cell, and cell-cell interactions, and 3D cell culture models, animal models as well, using biofabrication, human stem cells, disease modeling, drug screening, and also gene therapies to achieve the goals. So recently, we kind of summarizing how we can use the stem cell from the current stem cell to for the cardiac tissue engineering. So we're able to use the human pluripotent stem cells to identify or differentiate cells in the heart, including many cell types already mentioned, such as cardiac fibroblasts, immune cells, cardiomyocytes, endothelial cells, and also smooth muscle cells. We fabricate them using different engineering tools, such as the micropatterning, engineered heart tissue, decellularization, scaffold, free aggregation, electrospinning, and also 3D bioprinting or hydrogels. Then we're able to fabricate those cardiac engineered constructs. Use those constructs, we're able to do regenerative medicine or therapies or disease modeling and also drug screening. From there, we kind of, we talk about a lot about iPS cells. We kind of did our same using centivirus for the Yamanaga factors to reprogram the cells from the somatic cells, and we're able to generate those colonies of the iPS cells after 2 days, 14 days. And then use the cells, we're able to identify the markers, which is those 4 markers and beyond, and also we did teratoma formation to identify those germinal formations, mesoderm, endoderm, and mesoderm as well, ectoderm as well. So use those cells, we're able to differentiate them into cardiovascular cells, including cardiomyocyte and also endothelial cells. So one interesting question I would like to ask, because when we compare, or the other one also compare that the stem cell derived cardiomyocyte, they actually are really immature. So compare the transcriptome profile from the 2D cell culture at day 20 to day 60, up to able to compare them to the human fetus heart or human adult hearts. You can see that the transcriptome are growing or changing from embryonic to adults from those cell types. I mean, that's the cardiomyocyte derived from stem cell. Even day 60 in 2D cell culture models, they are so immature compared to adults or even not close to the fetal heart. So from there, we're able to climb this pyramid on the right to show that, okay, maybe potentially we can increase the maturity by looking into a calcium transient sarcomere formation, and also looking into the shape of cells, which is mostly looks like adult cardiomyocyte. So the idea is how can we make the stem cell derived cardiomyocyte into this rod-shaped cardiomyocyte in adult. So then we want to go back to see the hearts itself, so which is kind of really interesting, beating, fascinating organ in our body. So from there, we can see the anatomy of the heart tissue. We're able to see this muscle fibers formation in the hearts, and we dissect further into this cells kind of histology, able to see the cells interactions or cells forming this intercallic disc of their, and we're able to see the cells actually forming junctions, couplings in one direction with electrical and mechanical couplings. By using gap junctions, hearing junctions, and also the other junctions as well. So from here, we can create this pretty simple micropatterning skill using this stem cell micropatterning for geometric confinement. We use this kind of stencil to pattern cells into, on the right, you can see there's a single cell pattern with a 10 to 70 aspect ratio. And then on the left, we're able to see the 10 to 700 micrometer aspect ratio to make the cell-cell connection formation into muscle fibers. And we're able to have 10 cells in one slot and to do the micropattern. From there, we form this hypothesis whether we can to allow the cells, the cardiomyocyte derived stem cell to have end-to-end connection or couple to form these cardiac microfibers. The cardiomyocyte will be subjected to a high maturity as adult-like cardiomyocyte. So we designed these experiments to show, okay, we're able to differentiate cells around day 12 and day 16. We're able to replay the cells onto this hydrogel, which is around 10 kilopascal, around physiologic stiffness. We're able to have these 4 different groups, the random single, random cluster, and the single pattern, and also micropattern ones, which is the cardiac microfibers, ICMF. From there, we culture them up to day 65 and able to examine the sarcomeres contractility, titular formation, calcium transient, cell junctions, and also marker gene expressions. So from there, it's pretty obvious we're able to find our sarcomere structures by staining the troponin T and also alpha-actinin. Here we're showing the Z lines by alpha-actinin. We showed alignments and also increasing the regularity by the FFT measurement. And then we're able to see that from micropattern cells regarding the single patterns or ICMF, they have a similar, the highest length of the sarcomere length, which is around 2.0 or 1.9 micrometer. From there, we're able to measure contractility by the video-based pixel displacements, and we're able to see those contraction of the cells from single ones, random clusters, and also the single pattern. Further, we look into the ICMF. It has pretty nice speeding and also has really nice synchronized speeding into each microfibers. They are not connected, meaning that like you can treat them as each independent microfibers of the ICMF. From there, we measure the pixel displacements to represent contractility and able to see those functions and able to compile them among the 4 groups. We find out the contraction velocity and also contraction relaxation velocity in the ICMF, they are the highest compared to the other groups. So this meaning that after micropatterns, the cells actually were forming pretty nice sarcomere structures and also have the highest contractility. From then looking to the calcium transient, here I just want to mention a little bit about the titular formation. So this is a mouse ventricular cardiomyocyte, adult cardiomyocyte. You can see pretty nice straight structures of titular formation, mostly look like colloquial with Z lines. From there, it seems like the tubule should be forming like this. And this is kind of the animation of the cardiomyocytes, and this is a tubule formation inverted into these cells by extension of the cycloma. And we're able to see that the calcium transients will be happening along with the tubule into the cells and to trigger the contraction of the myosin and also actin. So from then, if we pattern cells are able to see the calcium transient like this, and also you can see the calcium transient into each individual ICMFs. From there, if we imagine, okay, we have this nice formation of tubule in adult cardiomyocyte, and also if the tubule formation not mature like this, we block this area and we measure the calcium transient, and you can see what happens, right? So you can see the difference. Maybe you can see the calcium transient will be not exactly the same in the adult cardiomyocyte compared to the immature cardiomyocytes with blocking or not really nice formed tubules. So from here, we can imagine that the calcium transients will be supported by T-tube formation to have the synchronized calcium transient or even calcium transient inside the cell from the outside or the out layer to the center parts of the calomel. So we did a measurements on the calcium transient of different cells. So we're able to see on A and B. From A, able to see a little bit different to the B on the calcium transient. If we profile the edge and also the middle parts, C is representing the B. You can see this pretty much overlap between the edge and also middle ICMF. And you can see the calcium transients like a straight line. If you see the D profile from the A, you can see the edge of the cluster random compared to the middle of the cluster random. The transient of calcium, there are slight difference or misaligned, meaning that's like forming kind of in synchronized or not in even distributed calcium transient compared edge and also middle part. So we define them. So it's from this straight line to the V or W shape, we calculate or count the cell number based on the calcium transient profile. We can see that in ICF, we have over 69% of cells out of 80 cells. They have in a straight line shape and compared to the micropattern singles or random clusters, they have in last 48%, now 39%. From there, we did a little bit more studying about T-tube formation. We're able to see straight structures from this ICMF compared to a random cluster on this kind of on the T-tube formation. Now, this is the stain by the cell membrane structure, and further we stain with the specific markers of the covalent 3 to show that the T-tube formation, which is co-localized with Z lines and in the ICM group compared to the single pattern ones or random clusters. So also we calculate the sample numbers. We're able to see the similar structures in the ICMF over 10 of them, and also 5 of them, they are not straight structures compared to the micropattern single ones. You're able to see much less cells having this straight structure of T-tube formation. Then we look into the cell-cell interactions, which is coming back to the hypothesis that if we're able to pattern cells into microfibers-like structures, we're able to control the cell-cell junction formation. We're able to see this junctions formation, which is the gap junction in green, to show this is end-to-end connection of the cells compared to random clusters. So they're kind of having this junction all over the cells in 360 degree. And also this is more like oriented or in one direction of the junction formation. Also, we're able to see the adherent junctions in red in the ICMF compared to the random clusters. So we further want to do something really interesting, and also I have to say like pretty brave to do that, because if we consider the normal heart's beating or animals or human hearts, the beating rates is around 0.5 hertz up to 1.5 hertz. So if we beyond that or lower than that, it's really not a good way to examine the hearts inside of the body. However, by using the stem cell derived calomelcytes, we can do a lot of testing on them, including this one. So this is probably the first time we're able to do the electrical stimulation challenge on the cells. We stimulate cells continuously from 0.5 hertz up to 1 hertz, 2 hertz, and up to 7 hertz. So typically, you won't see a lot of things from 7 hertz, but actually you can see some things from here. So we do measurements on the calcium transient and able to see the 0.5 hertz, 1 hertz, and up to 5 hertz for this one, and also the lower ones able to see 7 hertz of the calcium transient. So you may notice a difference, right? So the question is, we are able to see a difference between the top ones to the bottom ones at 5 hertz. So you can see the frequency, if you just look at by eyes, you can see the frequency of the 5 hertz actually is pretty similar to the frequency of the 3 hertz of the inputs. But for the lower ones, you're able to see pretty much similar or matching of the frequency change, or you can see more frequency of the calcium transient with at higher frequency of the input stimulation. So we did a bit calculation to see these intervals and also inputs of hertz to show that, okay, they are kind of matched at 0.5 hertz, 1 hertz, and 2 hertz, up to 3 hertz. But up to, if you look at 5 hertz, actually it has pretty much a higher interval, which is different to the inputs of 5 hertz. If you look at lower ones in the figure F, you can see this interval from 0.5 hertz to 7 hertz. You can see pretty 1-to-1 match of that. We did the relative percentage of the variation of inputs to outputs, we're able to see the lower, like 10% or 11% of the variation from 5 Hz, 0.5 Hz to 3 Hz, but up to 5 Hz, you're able to see a huge variation happens. Compared to the G, it very noticed the variation around like 10 Hz over the time. I mean, that's the ICMF has shown a pretty good calcium transients upon this pretty rigorous and also continuous electrostimulation on the cells to show the maturity of the cells. So overall, we're able to demonstrate that, okay, we have the hypothesis is cardiomyocytes are end-end connected or coupled, able to form these cardiac microfibers. The ICMF will be subjected to high maturity as the adult cardiomyocyte. So we test different groups, able to show that, okay, we able to see the sarcomere length alignments increase, contractility increase, electrical stimulation is more responding to the different frequency of the stimulation. We're able to see the calcium cycling increase and also T-tubular formation and also junction formation in this model. So this is pretty kind of proof of concept to show, okay, we're able to achieve this goal by micropattern cells into ICMF to show the maturity increase from there. So further, we're using kind of different cell type to show this model system. So one interesting disease or quite important disease we're showing here, we are looking to hypertrophied cardiomyopathy, which is the HCM. So usually, you can see 1/500 population have this type of disease, and you can see a lot of complications or phenotype from that, such as the arrhythmic sudden death, and also heart failures, atrial fibrillations. And also, it's pretty much you can see from right image, you can see the patients with HCM, they have pretty thickened ventricular septum. So one interesting mutation we're looking into, which is the myosin binding protein C3, which is the mutation counts over 20 or 25% mutation up there. So with pattern cells with different mutations, we got the cell line from Glass Institute, and also with thanks to Dr. Ma and also Dr. Nate, the last thing. So we're able to pattern cells with different genotypes with the wild type and also heterozygous ones and also homozygous ones. And we're able to measure this contractility from those cell lines. This is pretty kind of comparison between different days of patterning and also able to show the different cells with different mutations to see the contractility difference of them. So we're able to identify the homozygous ones shows more obvious kind of difference, contraction velocity and also amplitudes, and also the TTPs, relaxations, which is more looks like we can see from hypertrophic cardiomyopathy patient. So from here, this is kind of hearts. We dissect into kind of from muscles into the single cells or cell-cell interactions. However, we notice is this kind of 2D models, and our heart is like more 3D structures. So from there, we want to see whether we can find out something related to the 2D model towards into the 3D model, and also we're looking to the cell types, which is we're able to find multiple cell types in the human hearts. So meaning that our heart is like chamber formation, is beating all the time, and also able to identify the multicellular structures in the heart. So achieve this goal, we want to model this by this organoid. So I've probably already heard about lots of organoids in the past 5 or 10 years. So this is kind of summary with this in the book chapter to show how we can use this human stem cell, pluripotent stem cell derived 3D cell culture models to model endocardium and to differentiate them to the ectoderm and the mesoderm and endoderm into different organ differentiations into organoids. So use those organoids, we can categorize them into the structures, compensations, functions, and also even in vivo transplantations. So we can see the application of them. We can use the organoids for disease modelings and pharmaceutical discoveries, including late onset and also developmental disease and also high throughput screening organ chip system. In the future, actually now a lot of people doing the vascularized organoid and also assembloid as well. So in here, I want to introduce a little bit about the vascularization of organoids we are doing in the past few years. So we kind of generate this direct differentiation cardiovascular cells into the organoids, but we generate this triple recorder cell line to demonstrate the different cell types in the hearts, including the cardiomyocyte in green, and endothelial cells in orange, and also the smooth muscle cells in the CFP, which is blue. So using these cell types, and also we're able to differentiate them in and also form a pretty nice protocol, including the different small molecules and also growth factors to differentiate those kind of cells, endothelial cells, smooth muscle cells, and beyond that, in one batch into the organoid. So from this one, we also use these micropatterns to differentiate cells into these cardiac organoids. We want to vascularize it into the VCOs, vascularized cardiac organoid. So from there, we differentiate the cells into these vascularized organoids, but we compare different conditions to optimize it, which one with combination of growth factors and small molecules give us the highest expression of the green, and also high expression on the orange, and also high expression of the CRP in blue, to show that, okay, we're able to show the most vascularized cardiac organoids from the differentiation. So we identified condition 34, which is the optimized condition for VCO formation. So we did a little bit RNA-seq at different time points of differentiation to show that from day 0 up to day 16, we're able to see the pluripotent stem cell differentiation day 0 up to mesoderm at day 2, and also able to show the cardiovascular progenitor cells at day 5. And then we can see the cardiac genes, and also smooth muscle, and also fibroblast gene expression starting from day 8, day 16. We're able to see endothelial cell genes, which is more highlighted or overexpressed in the day 10, and also day 12, and day 16 in the VCMs. So we use lifestyle imaging to identify this vascularization formation in the organoid, and to show that from day 2 up to day 16, we can see nice formation of the organoid with vascularization, and also forming this kind of myocardial ring-like structures in green of this organoid. So we measure these functions of the organoids by staining them with the flow holes to show the memory potential, and we look at the contractions and the beating rates, and also looking to, we're using the isopropanol, which is the ISO to trigger cells for contraction. We see pretty nice increasing concentration along with the beating rates in the VCMs. However, we don't see that in the controls. Controls is just cardiac organoids without vascularization. We also measure the NO secretion, which is the representing the endothelial function of the nitrate secretion, or nitric oxide of secretion. We see the increasing nitrates in the supernatants of the VCO organoids compared to the control groups. So we did single cell sequencing to identify the cell types from the VCOs, and also compare with the in vivo human fetal hearts at 6.5 post-conception week. So this is published data from 2019 Cell paper. So most of the cells we're able to identify from these fetal hearts, we're able to identify them in the VCOs. Only difference is that we couldn't find the similar ratio there. So that's something we are doing to improve those ratio to match what we can see from cell types in the fetal hearts in our VCO model system. Further, we're able to identify subgroups of or subtype of cells in the organoid by looking to the single cell sequencing data. So we're able to identify the ventricular cells, atrial cells, and also venous, and also arterial and endothelial cells as well, and also fibroblasts and the epicardia, and also small muscle cells in those clusters. So we still want to find out whether the cell instructions are matching or close to the cell instructions in the fetus hearts. So we're looking to put in famous Notch, delta, JAG interactions between those 3 different cell types. And most of those interactions we're able to find in the VCOs, we can find in the fetus hearts. However, there's still many of them are missing, including Notch2, DL1, also Notch2, Notch3, and also Notch1 in different cell types. So we are still looking to the way to improve that by enhancing those cell interactions in Notch signaling pathways or beyond that. One interesting way we can use the organoids is to study the congenital heart disease or model congenital heart disease defects. This is something we are looking to the disease, which is the HLHS, which is hypoplastic left heart syndrome. So this type syndrome is pretty severe ones. We can see that syndrome from 1/4,000 newborns, and also it's pretty fatal. It results post-surgery of the born. So we were able to see a lot of other developments of left ventricular, mitral valves, aortic valves, and also ascending aorta. So most of the times we're able to see deficiency of the cardiac vasculature structures in those hearts with limited blood flow and also heart muscle and chambers as well. When we look into the genetic etiology, we're able to see those kind of mutations relevant to NKX 2.5, TBX5, and also EST1, and also most of the times we're able to see some mutations relevant to Notch1. So that's why it's kind of interesting way to model this disease by looking to Notch1 signaling pathway. So we use small molecules at this moment to inhibit Notch signaling. So this is inhibiting all Notch pathways, not including Notch1, 2, and 3. So this is not specific. However, we're able to find something by using these steps to inhibit the gamma secretase function. If there's a gamma secretase inhibition inhibited, we're able to see the NICD will not be released from the coupling between signaling cell and also receiving cell of the ligand of the Notch ligand and also the Notch receptors. So meaning that the nuclear translocation of NICD will not happen, and we won't see this following target gene expressions from there. So by using the Notch, we're able to measure the fluorescence change over time by starting day 2 and now up to day 16. We're using 2 different concentrations to inhibit this Notch pathway from 1 micromolar to 10 micromolar, and able to see, okay, this endothelial cell differentiation developments is definitely inhibited at the Notch signaling pathway inhibition. So we measure the area of the fluorescence to see the depth in decreased endothelial cells developments in the organoid. And also we look into this cardiomyocyte differentiation and ring structures with the control group. However, we don't really notice much about differentiation from 1 micromolar and also 10 micromolar. When that steps not just inhibits the endothelial cell developments and also inhibits the cardiomyocyte developments over the time. So that's kind of inclusion we want to mention that in the scientific and also technical advance of iPS technology for studying the cardiomyocyte and endothelial cells developments and also heart development disease as well. So with pretty normal, simple micropattern technology by stem cell micropattern to mature the cardiomyocytes into ICMF to show the enhanced segment structures, contractility, calcium transient, and junction formations and electrical stimulations, and also gene expressions. So identify these kind of cell types in the VCOs and also the Notch canonical pathways is significant upregulated in the vascular organoid. However, we're able to inhibit those Notch pathway to lead the EC and cardiomyocyte disruption differentiation. So this could be a good model of the VCO to model this congenital heart disease, looking vascular structures and also cardiac structures formation there. So this is kind of the presentation, and also like to thank my group. They're lovely students in the lab. They're doing a lot of studies in the past 2 years. They're all supported by this startup from UNT and also NIH and also the Harvard Small Heart Trust. So that's everything. Thanks.
Thank you, Dr. Yang, for a nice presentation on cardiac organoids. Now it's our discussion time. I encourage everyone can turn on your cameras, so we have a really interactive interactions and discussions. Before we start, I would like to introduce our panelist, Dr. Jing Guo. She is currently a postdoc scholar at University of California, Berkeley. And before that, she obtained her PhD degree from the Department of Mechanical Engineering and Material Science in Washington University and St. Louis. Her postdoc, her doctoral research focused on investigating mechanical overload to have an effect on iPSC-derived cardiac tissue, particular focus on the MYBPC3 mutation proteins. So the the procedure will sort of like I probably like can initiate some questions to all the speakers, and also Dr. Guo also can help facilitate some discussions. At the same time, I also encourage the speakers. You're welcome to also ask questions to other people as well. To begin, I'll probably start with a question to Dr. Bao. So I'm not a immunologist or any immunotherapist field, but I'm just interested. I know you have this card iPSCs, right? Basically, you can differentiate into any immune cell types that you want with the card presentation on the cells. I know you sort of focus on the NK cells, but and you also did some research on T cells. Now you're moving a little bit into the neutrophil. I'm wondering, have you ever tried to do the injection of combination of two different cell types instead of just focusing on one? Do you think there's a benefit, or that might complicate everything?
Yeah, that's a good question. And we co-inject, first of all, CAR NK and the CAR neutrophil into mouse. Due to the old time, I skip my results on that slide. There's no kind of obvious combination therapy effect after CAR NK or CAR neutrophil injection. So that's one we already know. And the reason why we didn't use kind of the common use CAR T cells, because the differentiation is still not so well reproducible. Although there are already many protocols or papers on CAR T cells from human stem cell. Whenever we apply the protocol to our kind of cell line, it sometimes doesn't really work out. So we can get some, but for mouse study, sometimes you don't have the high quality or sufficient number. We were just not doing any CAR T on animal study. But I mean, that's a good point. And we may later on, and actually we are doing some work to use just the primary CAR T cells using the standard one, using lentiviral or other to make the CAR T, and trying to see the combinatory effect with particularly focus on CAR neutrophil and CAR T cell.
Just follow a quick technical questions about the neutrophils. So I heard, I never do it in my lab, I heard that neutrophils are really hard to culture. Especially you isolate the neutrophil from blood. So they don't really attach, right? So I'm just curious, is there any difference from iPSC derived neutrophils?
Here's the thing. The reason why there's kind of nobody doing CAR neutrophil in primary neutrophil, there are people doing CAR microphage already in Nature Biotech a few years ago. The reason is the neutrophil, they are one bad thing is they are the most abundant white cell in circulation, but they are very short life. So mostly maybe die in 1 or 2 days. So it's a very kind of refresh almost every day the neutrophil. So that's why we using kind of stem cell, because you can differentiate and get all kinds of neutrophil progeny cells or terminally different neutrophil, and there's no issue. When you put a CAR into stem cell, you can get a CAR neutrophil, but if you put a CAR in neutrophil, they may die right away. So that's kind of the technical barrier. And in terms of blood or immune cell culture, it's more, as I mentioned, suspension culture, and there's no huge difference. But in general, a stem cell culture is relatively expensive, but primary immune cells are also expensive because you need to supply many of the cytokines into the media. So that's kind of also expensive to culture. But other than that, it's very similar. You can put the cell in just like a 96 or whatever well played, or you don't have to like using a shaker to culture. But for sure, if you want to go to larger scale, it's better have some like a shaker or like a small bioreactor to do that with kind of like a stir. But for most like mouse or less animal study, it's a fine. Suspension culture is similar to like a stem cell or attached cell culture.
Thank you. Other questions?
Maybe. So I have two questions. Firstly, I want to thank Dr. Ma for the invitation. And also I want to say congratulations to all of the speakers for great presentations and great work. I really enjoy learning the research using the microfluidic device to study the developmental biology and also using the iPSCs to engineer CAR T cells to test in vitro and using mice and to use micropatterning technology for iPSC, cardiomyocyte heart pathophysiology studies. So I have two general questions for all of the speakers. So first question, I'm just curious, what are you guys' insights on steps that need to be taken in order to increase the translational potential of stem cells, specifically the human iPSC, for potential preclinical and clinical applications, and whether you guys think in vitro models can eventually replace some of the in vivo models? And also, second question is more about general career advice on young scientists. So I'm curious to hear your career path on how you guys get started in this field and how you guys find your specialty and expertise to open your research lab and to get to where you are right now. Thank you.
Maybe we can start with the first questions. Across the three speakers, and then we move to the second one. Who want to get started with future stuff for the iPSC based either therapy or in vitro models?
I can start it. So I believe everybody here is doing the kind of same thing for promoting the iPS-derived products for in vitro modeling and also in vivo therapeutic purpose. So definitely, this is kind of future of the science or future of technology when used for screen drugs and also find really in vitro therapeutic treatments for the human diseases. So right now, a lot of people still question us whether this is similar to animals, right? So a lot of people were questioning, especially Revere 3 always question you, why don't you use the animal model to verify your system? But most of the times, we are trying our best to really identify those cell types, pathways, signalings, and also cell interactions, morphology, structures, and functions to prove that, okay, we can benchmark most of everything from this in vitro system to match what happens in vivo, especially close to human being, human organ tissues. So, but still a lot of way to go, right? So maturity is an important thing, and also a lot of in vitro environments, which is not exactly the same as we can see from in vivo. Even though we are creating a lot of artificial organ or tissues, they're still not the same as we can see from the organ or tissue we are having in the body. So that's a lot of things we can do, but also we are doing our best to create those systems, including myself, we are more focused on the hearts and also more focusing to using different engineer system to mature the hearts and also make the hearts more looks like the human hearts, including function structures. So that's what we are doing.
Maybe Dr. Zheng, you can follow up with the in vitro model part.
Yeah, for the in vitro model part, my opinion is I don't think these in vitro model eventually gonna replace animal models, but it just we have another tool or another perspective for our research. Let's say for drug screening purpose, usually people do, let's say, in vitro culture cells and the small animals, large animals, and all the way up to human. But before we start conducting the very expensive, very long scale, large animal or human test, we want to accumulate as much information as we can. And the in vitro stem cell model just give us another tool to gain some insights to enhance the drug safety and efficiency. That's my opinion. And another question as to the limitation of these stem cell technology. Well, as you can see in today's talk, all the three speakers are from engineering department. I think now, again, still my personal experience, all the organoid or in vitro model of used stem cells, they are not that reproducible to a certain degree. It depends on the skill of the person who's really doing the experiments. And I think that is a huge limitation right now in this field. That's why it requires a lot of engineers to develop these tools, either micro devices, micro patterning, or new way of generating CAR T cells, just to make it more reproducible and controllable, so the results are more trustworthy. I think we are. I think in the very beginning of stem cell research, it's really heterogeneous, but now it's getting better since more engineers are devoting their efforts in this direction. I think that is certainly a very promising field, but we still work has to be done.
Great, thanks, Dr. Zheng. And Dr. Bao, like we all know immunotherapy is a really hot area now. So can you share your insight how iPSCs can be used for future cell therapy, not necessarily in the immunotherapies, but like in general for therapeutic applications?
Yeah, I think Dr. Yang and Dr. Zheng has provided really good perspective on the engineer or the kind of drug screen side. And from more a cell-based therapy, I think everyone here already have heard there are kind of not many, but already several or a few clinical trials using stem cell-derived therapy, using heart muscles to treat heart damage in clinical trial, I think in Japan. And there are some diabetes or some neuron clinical trials already ongoing, by one of the kind of few funding a company Blue Rocks, I think one of the kind of funding companies that are using stem cell derived cells. And I don't think there would be any issue in kind of clinical translation when you have really good or high quality cells. And in the immunotherapy field, I think it's even a less barrier in clinical translation because people or clinical docs already translate all kind of hematopoietic stem cell transplantation or CAR T, CAR NK. There are lots of a million, not a million, but at least hundreds of clinical trials all around the world. And in terms of stem cell derived CAR natural killer cells, and there may be some other already in clinical trial, but one of the famous companies, Fate Therapeutics, which kind of already have I think 8 or 10 clinical trial when people they make iPS derived NK cells. But they have disclosed a clinical maybe 2 or 3 phase trial. There's no a huge benefit of CAR NK. But I don't think it's an issue of CAR NK from iPS. But maybe there are an engineer approach to engineer the CAR, or the manufacturing for NCAR may not be the the best, or the major barrier in I think iPS based cell therapy. One is maybe how to get a good cell population for transplantation. That's maybe the key issue the field need to address before more kind of a clinical trial or more I think a therapeutic benefit can be achieved. not necessarily you have to make the cells very mature for transplantation purpose, but you may need a balance, kind of the say engrafter efficiency, the survival, or kind of the maturity. There are lots of to consider when people trying to translate iPS-derived cells for clinical application. And that's my kind of thought on this. I feel, still very promising on iPS-based therapies, but again, there are still need some barrier need to be kind of overcome.
Thank you so much. And now maybe we can circulate our next questions. So within the time limit, maybe 3 speakers can share some advice how to pursue a successful academic career path. We'll get started. Dr. Bao, maybe you can start. You can just continue.
Yeah, I can continue. This is a really tough question, and I think every kind of faculty have their own career development path. And my experience is in general, on average, engineer department is relatively have more openings or less competitive as compared to a pure biology department. In terms of iPS research, and the training path, or we all can claim our as multidisciplinary researchers, but actually it's sometimes more where you kind of want to find your a department or affiliation for your job. I think one easier way, for sure, everyone need to have great work done in impactful work research from their PhD training or a postdoc. That's kind of the maybe priority to get you kind of get to interviews, to get interviews or on-site interview. Secondly, you may need to kind of with your kind of package, you may need to expand some other skills, say presentation skills, or communication with the faculty in other area, or in general, this kind of more technical skill set that may not necessarily associated with your ability to perform research. But maybe at your postdoc period, you want to develop or spend some of your time to trying to increase this skill set. And that's mostly, for maybe the job interview tips. But I think after establishing your lab, I'm still young in the field, and but one thing I learned is sometimes you need to be more patient before you kind of compared to your postdoc or your own PhD training, because now you need to be the leader, and when you begin, you may need to do research and also train people at the same time, and also writing and teaching. So maybe just be patient to think what's your kind of priority of your research project, and also your time management. What kind of you want to assign more on your when you begin your a career in faculty position.
Thank you, Dr. Bao. Maybe Dr. Yang, can you share of your opinions?
Yeah, I'm happy to share with something I learned in the past 2 years, because I'm still probably, because I think Dr. Bao has covered a lot of things there, and also has been doing this great job in the past, I believe, 4 or 5 years in your position. So definitely I'm learning from everybody else at the same time to build up my career, and also to see something I can do in the next 3 or 4 years to get my tenure. But anyway, so I think there's a lot of things, you need to start prepare really early, as sometimes even in the PhD program, and later on in post-fellow program. So that's kind of your foundation. You have your expertise and also something quite important to establish upon from there to have extend this expertise when you become independent. So in my area, we're in pretty competitive areas. A lot of people doing hearts and also cardiac tissue engineering. So that's why I try to find a lot of collaborations within area and also distance areas as well to see whether I can really do something together. Because right now, no one project can be done by just one single person. So you need to really find a lot of collaborations, and also you can identify some really good collaborations. So you don't want to say you want to collaborate everybody else, or so there's no way you can do that. But also you can see depends on those people's personality, the way they are doing things. You're able to find out, okay, could be a lifetime or at least for long-term collaboration. And from when you engage with them, talk to them. So it's important. You know, as a junior faculty, it is pretty difficult for us to really reach out to people. They didn't know you. Like you are pretty young, so you didn't really have much to show to the other one to maybe benefit the other one's research. Meaning that you need to do most of the job by yourself. Such as writing grants, including senior professors, but I need to really find out that you are useful for the other labs and for potential collaborations. And also you need to identify those directions could be really beneficial in the next 5 or 10 years. So I'm lucky in this DFW area, so we are able to find a lot of people in this area, and also including school medicines and also College of Engineering at different in universities. So and also try to meet people in a conference, to be kind of target those people. You read those papers, you identify those corresponding authors, first authors, rising stars, and also senior professors. So that's kind of my suggestion. But also take your time to do things now, so you may be able to achieve something in the next 2 or 3 years, and then publish some papers and also get some grants. Gradually able to publish bigger ones and also get bigger grants, and then you're able to establish your career gradually from there. Yeah, that's my opinion.
Thanks, Dr. Yang. Maybe Dr. Zheng, you can maybe take the last couple of minutes.
Yeah, thanks, Dr. Yang and Dr. Ball for the very good suggestions. I think they pretty much cover all the important aspects of being a young or junior faculty. Again, I'm still at the very early stage of my career. I just want to say based on some of my own experience or my observations of some very successful young colleagues, is that in the beginning, it's a very hard job. But my suggestion is you don't think that much. You just do your own research. And if you get something unique, you contribute to the field, and you will find the collaborations, the finding, the students, the resource will naturally come closer to you. So of course, it's a very challenging job, but again, do not be too nervous or do not doubt in yourself. As long as you get your work done, you did everything like Dr. Yang and Dr. Bo just suggested. Eventually, you get tenure and you will get there. And another thing I want to say is if you want to pursue an academic job, you need to plan very early during your early PhD years. And at that time, you need to know that research in nature is a risky path. Because we do research, we don't know whether it's going to be successful or it's going to be impactful or not. What I want to say is do not always stay on the safe side. Do not always stay in your comfortable zone. Sometimes you need to take the risk and really identify a good direction and just stay there, be patient, eventually make your own contribution, and the field will recognize you naturally. They will just take you. And of course, other than the research, you need to learn how to present the work, how to communicate with people, how to expose yourself to the community, like all these things. I think that's I want to say here.
Okay, thanks, Dr. Zheng, and I want to thank everyone for this sessions to make your great presentations, and it's a great discussions. Thank you all. I'll see you in the conference. Bye.
Thank you. And
Thank
see
you.
you, everyone.
Bye.
Bye-bye.
小船是不是忘了介绍那个了？下一期的了。
不再是奇迹，不再是幻想，此刻真感觉全世界对我鼓掌。不必太在意身旁惊奇的目光，可以点点头，可以放声歌唱。我创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞。不再为曾经失败放弃或感伤，努力才是真的方向。I can， 没有什么可以阻挡心中无限的力量。I can， 你也能够像我一样飞越最高山岗。I can。没有什么可以阻挡心中无限的力量。I can， 你也能够像我一样飞越最高山岗。创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞。不再为曾经失败放弃或感伤，努力才是真的方向。I can， 没有什么可以阻挡心中无限的力量。I can。能够像我一样飞越最高山岗。I can， 没有什么可以阻挡心中无限的力量。I can， 你也能够像我一样飞越最高山岗。能够像我一样飞越最高山岗。
祝你生日快乐！该吹蜡烛了，来，我帮你。啊！祝你生日快乐！生日快乐！
好漂亮的花，我很喜欢。
这是我的礼物，多层陶瓷电容器，